Assalamu alaikum my dear students, I hope you're all fine. Today I am here with another informative video about the food tests, which is a topic of IGCSE and GCSE Biology O levels. It's from the chapter Nutrition. My name is Adnan Khan. Students, as you know that this topic is being frequently asked in the previous papers as well, and it is most important topic among the most important topics in paper 1, paper 2 and paper 6 of the O levels, and also in the ATP of the IGCSE. So let us discuss different type of food tests that we perform. As you know that the food samples may contain different sort of nutrients, and upon them we apply different food tests to find out their quantity or to find out their presence. Now in this video I'll be discussing the 4 types of the food tests. Number first, we are going to discuss the Benedict Test, which is a test for reducing sugars. Number second, we are going to discuss the Buret Test, which is a test for proteins. On the third, we are going to discuss the ethanol emulsion test, which is for the presence of lipids. And the next, we are going to discuss iodine test, which will be for test for starch as well as test for starch in leaf. First of all, the Benedict test. The Benedict test can detect the reducing sugars in a sample. If you are being given a food sample and you are in doubt if it contains reducing sugars or not. So reducing sugars is a separate topic, but remember that monosaccharides discussed in your syllabus, all 3 monosaccharides, the glucose, fructose and galactose, they are all reducing sugars. The disaccharides that are more formal to you, the sucrose, maltose and lactose. Amongst them, sucrose is a non-reducing sugar. All other disaccharides are also reducing sugars except sucrose. In order to change sucrose to reducing sugars, you should treat it with HCl, and then it changes into reducing sugar, and then you can apply Benedict test. So keep in mind that sucrose is not a reducing sugar. So if you are having a sample and you are in doubt that if it contains reducing sugars, you should go for the Benedict test. Now the procedure of the Benedict test, students are being given the free end questions to write about the procedures. First of all, the Benedict test, we are going to add reducing sugar and heat with the Benedict's reagent, and the result, if the reducing sugars are present, insoluble red precipitate of copper 1 oxide is produced. Now what's the procedure? First of all, you add 2 cm³ of sample in test tube. Remember that the sample must be in liquid state. If you are going to apply this on some fruit, you should actually extract its juice and then apply it on it, and you should write about the quantity as you are taking equal amount of sample and equal amount of Benedict's reagent. So in this case we are also adding 2 cm³ of Benedict's reagent, and then we are going to heat it in boiling water for like 5 minutes. Okay, now this will be called a water bath, because sometimes students are asked how to safely test for the presence of, so this will be a safety measure as well, because the open flame may be dangerous. So in the beginning, as you know, the Benedict's reagent, its color is blue. Even if you mix it with a sample, the initial color will be blue, and if it remains blue, no reducing sugars present. If very low quantity of reducing sugars are present, then the color will change. After heating, the color will change to green through if there are low, okay, so it's yellow, okay, and then brown. And if there are more reducing sugars, then you should label it as a brick red color, so it will be containing like more reducing sugars. These are the positive results. And if there are no reducing sugars, then you get the blue color. The next test that we perform is the Buret test, okay, and it is to detect the polypeptides in a sample. The sample must be in liquid state as well. So we are having to detect the protein in these food contents, and remember that you should be dissolving them into the test tube so that you get a liquid sample. And here's the procedure. You get a sample and then you add Buret reagent. You can also make the Buret reagent at lab by adding copper sulfate and sodium hydroxide or potassium hydroxide. So it makes the Buret reagent. It's already available. If you are having it, then you just add the Buret reagent to the sample in equal amount. If proteins are present, you get a purple color liquid on the top of the surface, okay, and the color changes, the color of the sample actually changes to purple color. Purple layer appears on the surface, and if you are having like negative result, the reagent will remain blue and there is no heat required. You just mix the sample and then you get the purple color layer on the top. But remember that the sample must be in liquid state, such as if you are applying it to the white of the egg albumin, etc. So they should be in the liquid state. Or if you are having to apply it on the peanut powder, you must put that powder into the water and then you apply it. Now we are going to discuss about the third test, which is ethanol emulsion test. This test is used to detect the lipids. May include fats or oil. Fats are solid at room temperature and we obtain them from animal sources. Oil is liquid at room temperature and we obtain them mostly from plants. Okay, so in order to detect the lipids in a sample, ethanol emulsion test is performed. Now there are some steps you have to follow in order to perform this test. First of all, as you know, lipid is insoluble in water. However, lipid can be dissolved in organic solvent like ethanol. So you first put the sample into the ethanol, okay, and then you just mix them together, okay, and then pour the solution into the water tube containing water. So if the white cloudy emulsion present, it indicates that lipids are present in the solution. The fourth test that we are going to discuss is about the presence of starch. As you know, different food sources contain starch. So to detect the starch, we apply the iodine test. To detect the starch in food sample or in a leaf that has photosynthesized glucose and stored as starch, so you want to detect the presence of starch, iodine test will be performed. Here, the food test, for example the starch test, the purpose is, in order to detect the starch, first of all grind the food sample and then you just filter out the sample and extract the solution. So you get the extract and then fill the sample to a spot tile, okay, of the white background. So when you put them in the tiles, just add few drops of iodine solution, okay, and result, if starch is present, then the food sample will changes its color to dark blue color, okay. That is when iodine react with starch, if the starch is present, okay. So here we got the results, and if there are no starch present, iodine will remain in its own color, which is brown color. And if you mix iodine solution with starch, you get the blue-black indication that starch is present. That's it for today. Thanks for watching, and please leave your comments and suggestions. Don't forget to subscribe and take care. Allah Hafiz.